So it's all yours, Paul.
Great. Well, thanks so much, Kate, for hosting me. It's really lovely to be able to talk to this community. I'm a huge admirer of a lot of the work that's ongoing within this community. I want to tell you today a little bit about our cell-free work. I usually start with a few kind of reminder slides for the synthetic biology community, so hopefully you don't mind. One context is that synthetic biology often considers DNA as some programmable language, and therefore it's a kind of a whole basis of one basis of the field is to work out how to reprogram living systems at the genetic level. And I guess one of the interesting things about life, and of course this is very much important for people considering bottom-up synthetic biology or build a cell, is that there are all these really amazing things about living systems, such like context dependency, evolution, adaptation, natural selection, being a major driver for living systems, and often driven through things like non-predictive random behavior. And then at the molecular level, the extraordinary ability of macromolecules to self-assemble and provide emergent properties. Then the sort of dynamics of the system being nonlinear and steady state. And then, of course, these huge multi-scale interactions that occur between the smallest electronic changes within photosynthetic systems through to whole organism behavior. And I think the living systems are so amazing and so complex that one needs to be aware of these issues when you're thinking about reprogramming them at the genetic level, because they do pose significant challenges. And I guess just for fun, I always point out that living systems are more like those sandwich wraps as opposed to circuit boards. And of course, some of these challenges have been overcome through this kind of systematic design framework. And it's a very interesting framework, which of course could be easily and probably is already being applied to building cell type modules in the build a cell community. And the idea here is the design, build, test, learn cycle, which I think everyone probably knows about and understands. And I think this whole core context of synthetic biology is really interesting for me, and it's something I've been passionate and pushing for quite a while now. And our most recent work has just come out in medical archives, where we pivoted our infrastructure within the London Biofoundry to become a frontline clinical diagnostic testing station for SARS-CoV-2. So I think Biofoundry is incredibly powerful. Now, having said all that, in a Biofoundry context, I've been very interested in working on prototyping, and that is a main kind of opportunity for a Biofoundry context. And one of the things that I really was interested in exploring was how we could use cell-free in vitro transcription translation systems to enable kind of synthetic biology workflows and applications. And I think what's really extraordinary about cell-free systems is that you're talking about the essentially biochemical extract from a living cell that has all the machinery to carry out the two major decoding events that happen in living systems in terms of nucleic acids, and that is the transcription step and the translation step. So the implication of cell-free extracts is that you can essentially put in a piece of DNA that you design, and you can then observe both the transcriptional production of a messenger RNA, as shown on the right-hand side or left-hand side of the slide in this little red curve. And you can also simultaneously watch the translation of that messenger RNA into proteins. It's a very powerful assay format, and of course has a lot of context for the build-a-cell community. Now, of course, they're not new. So cell-free systems have been around a long time. Started off, 1907, actually, which is over 100 years ago. But of course, more recently, I think it was used to confirm the function of the genetic code in protein synthesis. These are E. coli cell extracts. In 2001, Japanese colleagues reconstituted the transcription and translation system with all of the components that allowed you to essentially decode DNA in a test tube, which was a remarkable tour de force, and is, one could argue the primitive biochemical mixture for the essence of life, if you like. And then, of course, later also around the same time, there was this huge growth in synthetic biology and a lot of excitement around the opportunities of synthetic biology and thinking about genetics and genetic circuitry and DNA as genetic circuitry, and how do we exploit that. And at that same time, James Swartz and colleagues really started pushing the opportunities around cell-free synthetic biology with some amazing scientists, Vincent Narrow, Michael Jewett, who's an absolute tour de force in the field, Sebastian Merkel, Sven Panker, Richard Merrick, Caltech, very involved in these early days of cell-free systems, and to some extent our group. And I think the real excitement of cell-free systems is this massive array of potential application spaces that have been prototyped proof of concept ideas that are coming out from many different labs around the world. Very interestingly, for vaccine and therapeutic production, and also for distributed biomanufacturing, are two areas of very deep interest, I know, for a number of groups. One area that's really exciting, which has really been pushed by colleagues in Toronto and also at MIT and other places in Chicago, is the idea of these low-cost paper-based biosensors, which is really another exciting area. But also in my own lab, we've been exploring how we use cell-free systems for new chemical synthesis and some natural product discovery, and also for prototyping gene expression. But it's also been used for education and outreach, and also traditionally used for optimizing protein synthesis. So this is an incredibly powerful field, if you like, where you can really start exploiting the biochemical machinery of a living system in many different ways. So prototyping parts and designs. So my lab got into this a while ago, and the assumption was that what we measure in vitro, i.e. in a cell-free extract assay, had some representation of in vivo measurements. This is a key concept of cell-free lysate prototyping, which allowed you to explore in very high throughput contexts, in cell-free contexts, many different designs, part characterization, all sorts of different interesting opportunities to look at genetic circuitry and control, before moving into a living system where it somewhat takes a bit longer and is somewhat harder in some contexts. And the other beauty about cell-free systems was that they allow quite a lot of intensive computational modeling. So it's a very powerful framework for prototyping. This is the first paper that my lab published, which showed the relationship between in vitro measurements and in vivo measurements. And I won't spend too much time on this, but just to say that at the time, which was several years ago, we were quite surprised that we were getting reasonably good agreement between a cell-free E. coli extract and a living system, and this was exactly the same strain and the same parts and the same construct. And you can see we were looking at promoters, RBSs, and other parts, and we were getting quite good relationships between the relative strength of those promoters, both in cell-free and in living systems. So that indicated that there was quite a good deal of relevance to looking at cell-free prototyping. And then from this, we developed workflows that involved high throughput automation. And this is work that was published a year ago or so ago, where we were looking at applying the ECHO liquid handling system to do essentially nanoliter droplets, and looking at very low volume cell-free reactions of 2 microliters in a 384 well format, allowing quite a lot of assay space for cell lysate that could be produced quite easily from different organisms. So, for example, from 6 liters of cells, this particular experiment, we were able to get 50 mils of cell lysate, which would allow over 120,000 different cell-free reactions. And so, by using those platforms, you can start thinking about prototyping parts and designs in non-model organisms. And I was really intrigued by moving away from E. coli into other systems. And the rationale and motivation for that was that other extracts, other organisms could contain native regulatory machinery, sometimes very specific cofactors and coenzymes, and also sometimes the native protein chaperones for folding. This would allow non-model organisms the opportunity to be prototyped, the parts, regulatory parts, to be quickly assessed for organisms that have very few genetic components that have been characterized. and also might allow you to express specific proteins and enzymes in these native cell extracts, and also allows potentially unusual biochemical synthesis or natural products. So I won't go through this in great detail, but the first organism we studied was Bacillus subtilis, and we published this a little while ago. And what was interesting at the time was that this was the first cell-free extract that had been characterized for this organism. And this just shows the relationship between in vivo and in vitro. So if you look at that graph, the blue and the sort of orangey, reddish, purpley color are in, blue is in vitro, the orange is sort of magenta is in vivo plate reader, and then the green is in vivo flow cytometry. So again, parts characterized using different methodologies, both in vivo and vitro, showing reasonably good correlations. The second organism that we studied was Bacillus megaterium, and the rationale for studying Bacillus subtilis was that it's a very interesting organism for industrial applications, and people are very interested in acting as a cell factory. Bacillus megaterium is also a very interesting organism, potentially industrially, but it's extraordinarily slow to engineer. It's a very large organism. As you can see, the orange cells there are E. coli, but it has got a very interesting secretion system, and there are opportunities for grams per liter protein production using this organism. So it is relevant. But what was extraordinary is that there's so few genetic regulatory elements for this organism. So we set about doing a whole series of prototyping and characterization. I won't go through it in too much detail, but this is now the essentially all the genomic promoter, native promoter context for Bacillus megaterium's glycolysis and gluconeogenesis pathways, all characterized using cell-free, both at the protein level and the messenger RNA level. And then a tour de force by our collaborators in Germany actually characterized some of these parts in vivo, because this takes weeks to get good transformants for Bacillus megaterium. So this was a real big effort. But again, showing similarities between the in vitro measurements and in vivo. So very quickly, you could characterize a whole bunch of previously uncharacterized regulatory elements from a non-model organism using this approach. And then you could create part library. So this is an RBS part library. I won't go into any detail, but just to show again, you could create parts for organisms that had never really been studied properly in terms of genetic regulatory elements. Third organism we chose was Streptomyces, which is a soil bacteria. It's a very interesting, actinomycetes are a very interesting class of microbial species. There are about 150,000 natural products thought to produce. Many of our antibiotics comes from Streptomyces. There are some cancer drugs, and there are thoughts that we haven't really discovered the full potential or full plethora of molecules within these organisms. So the motivation here was, could we develop a Streptomyces cell-free system that we could use to maybe look at natural product discovery, or to look at some interesting antibiotic synthesis pathways. So we made a high-yield Streptomyces cell-free system. This took a little while, and I think there is a lesson to learn when you go into these non-model organisms. Streptomyces is an unusual organism in how it grows, but we made a very robust cell-free system, and then we were able to do some classic in vitro, in vivo correlations. Here we took some synthetic promoters that had been published in a paper in PNAS, reconstituted them within our cell-free system, and then just plotted out cell-free versus the in vivo measurements. And again, we're getting good correlation between our in vitro measurements and our in vivo measurements. And this is for promoters across 2 different Streptomyces species, actually. The mammalian cell lysates was the next organism that we started studying, and this was the work of Margarita Kopnitsky and Kiva Canavan, both PhD students at the time, and they set about developing a whole series of workflows and part characterizations for HeLa cell extracts. And so we were using a T7 promoter, IRES library, TCAS9 repression system, and an L7AE translational repression. And this is just an example of the data that one can generate, but it's already been published, so if people are really interested, they can go and look into that in detail. So building biosynthetic pathways. So one of the things that we set ourselves the task of doing, and unfortunately we have not been able to complete this, was to set about trying to reconstitute the oxytetracycline biosynthetic pathway in our Streptomyces cell-free system. Now, this is a very complicated pathway shown here on this slide with a whole bunch of different enzymes, unusual cofactors, really quite a demanding pathway, and clearly that was probably a little bit too ambitious at the time. Having said that, we were able to produce all of the proteins, and that's shown here. So these are all solubly expressed in our Streptomyces cell-free extract. And in fact, when we expanded the number of proteins that we looked at, we found that we could express up to 19 different proteins solubly in our extract. We could also make a non-ribosomal peptide synthase, which is a very large protein, greater than 100 kilodaltons, solubly in our extract. So we've set the ground and the foundations for now building upon that pathway. So we're working through to see if we can get any of those enzymatic steps to work either together or separately. We have made some progress where we've taken this little mini operon of 3.6 kb, which allows the production of melanin, shown here. And then we've also taken a slightly bigger mini operon, this 6.1 kb synthetic operon, which codes for the production of a couple of heme-like proteins, precorin 2, precorin 3, and urigin 3. And we've actually shown that we can make these proteins within a cell-free extract from this little mini operon, which is excellent. So obviously we're pursuing other types of biosynthetic pathways, but those are the ones that I wanted to share with you now. From that, we thought about trying to think about novel xenobiotics. So what do I mean by this? So we chose a natural product, and one that was everyone in synthetic biology kind of knows about, which is violacin. It's a very nice looking molecule, as shown on this slide. It's a natural product from a bacteria called Chromobacterium violacin, and it's produced by a very well understood biosynthetic pathway, the BioABCDE pathway. And what's interesting about violacin is it has sort of interesting biological activities as shown in that long list of either antiviral or antibacterial. And it's been thought of potentially as a scaffold that one could think about improving its properties as potentially as a therapeutic. There are two versions produced of it, the violacin and deoxyviolacin. So it's a very interesting test model. It's well understood. And it was interesting enough for us to explore this idea of whether we could make some kind of novel variations of it. So this is Hung-In Lei, who's now a postdoc who was in my lab as a PhD student. And Hung-In, working closely with our colleagues in St Andrews, this is a collaboration with Professor Rebecca Goss. The idea here was it could be used a mixture of both in vivo biosynthesis, in vitro chemical derivatization, and then produce compounds that were derivatized from violacin that we could use in some biological screens. So the concept here is that you build a biosynthetic pathway in a living system. This is with the ABEDC pathway, violacin pathway, that you then feed into that living system a whole series of modified tryptophans. These will be methyl, hydroxy, chloral, fluoro. And the idea was that those derivatives would then carry all the way through to produce new derivatives of deoxyviolacin here. Could you also exploit other enzymes that work on tryptophan? So in the Rebecca-myosin pathway, there are a couple of enzymes that will convert tryptophan into chlorotryptophan and feed those into this pathway as well to make other variations of the deoxyviolacin and oxyviolacin. And so that's sort of workflow here. So these tryptophan analogs, they're fed into this living organism producing violacin. The violacin molecules are extracted out using some organic solvent and then analyzed using mass spectrometry. The bromo derivative is then purified, and then that derivative can go into further in vitro chemical modification and to produce a whole variety of different analogs. And then the idea here is that all of these can be tested using a biological assay. So here's the violacin pathway. So VioA, VioB, VioE, VioCD. And you'll notice there are two derivatives produce the violacin and the deoxyviolacin molecule there. And both exist in the bacterium. So here we've got our tryptophan, and the idea here is that we will take all of these tryptophan analogs and we will feed them into this pathway and hope that they go through the whole biosynthetic transformation steps to produce these derivatized violacin or deoxyviolacin. Now, the good thing is that we know quite a lot about these pathways, but the first thing we wanted to do was to make sure that the substrates went through this first transformation step, which is essentially a proton abstraction to produce this imine intermediate. And the way to do that was to solve the crystal structure of the first enzyme VOA, which we did, which Hung-In did. And then, and soak in, this is tryptophan in the active site of this enzyme, and this is the FAD cofactor here. This is the key distance here. So this is the FAD, this is the tryptophan, that's the carbon alpha of the tryptophan, and that's the proton that's sipped off from the tryptophan to produce this imine intermediate. The active site, very well understood. So Hung-In took all of the derivatives that we were going to screen and soaked them into crystals and solved the crystal structures of all of these derivatives, and found that surprisingly, they all bound in a similar kind of position, but there was a certain amount of slop in where the indole ring sat. But what was really interesting was that the distance between the carbon alpha and the FAD was retained, implying that you could catalyze that first proton abstraction step. Now, we did some very simple Michaelis-Menten kinetics, and we found actually that this tryptophan is shown here, and then you look at the other derivatives, and although the kinetic is slightly different depending on the derivatives, they're all pretty active, and they all seem to indicate that you can get that first derivatization step done quite successfully. So that gave us a great deal of encouragement to say that if we can get it through this first step of derivatization, then the hope for this condensation reaction and then these other different steps, it was going to be quite possible to achieve the final goal. So we then decided to build a pathway. We made multiple variations, it's a classic synthetic biology type approach, different promoters, different RBSs, all the rest of it to try and get optimal yields, which we did. And then we fed in all of these derivatives. So this is a very, very busy slide, not meant for you to really read on any of this, just to say that there is a large array of potential violation and deoxyviolation derivatives that one could derive if this process worked. And what was really interesting is that it did. So here we've got these are pie charts, and what they're showing is some quantitative mass spectrometry data showing violation, which you produce through the normal pathway, deoxyviolation, and obviously tryptophan exists within the E. coli in natural metabolism, so you're going to be able to produce those two molecules anyway. And then we could look to see how much of the 4-fluorodeoxyviolation we're producing here, and the 4-fluoroxyviolation here. And so if you go through these, you'll see that there are different amounts of the different derivative deoxyviolations produced. And then if you use the ability to diversify is extraordinary, because of course you can do mixtures. So here we've got 6-fluoro, 5-hydroxy, 6-fluoro, and so on and so forth. And therefore you can end up producing a massive array of many, many different types of derivatives, which is what we identified. So we identified we could produce 62 analog derivatives in E. coli extracts expressing the optimized pathway. Then we made the chloro version, and we found we could make the 7-chloro violation, both the deoxy and the oxy versions. And this was quite important, as you'll see later. Now, one question we wanted to see was, could we make a bromo deoxyviolation? And the reason that that was important was that if we could make a bromo version of the molecule, then this would open up a whole series of different types of organic synthetic chemistry to us. And this is the work of Rebecca Goss's group, where she has applied the Suzuki-Nayora coupling approach, which is an intriguing kind of chemical cycle, if you like, that involves palladium. It involves a bromo group and also one molecule, a bromo group on one molecule and then boron on another. And then through this cross-coupling chemistry, one can produce a whole variety of different chemical derivatives. So this is Alan Obled in Rebecca's group, and he did this work. And what was really intriguing and remarkable was that he took the 5-bromo L-tryptophan derivatives that we actually managed to make, and then he would lyse open the cells, and then within the cell extract, he could carry out some very interesting cross-coupling chemistry, both within the cell extract and also purified. using these conditions and started to produce this first chemical derivative, which is this p-toluol derivative here, using this boron ironic acid here. And I think what was really interesting from this was that he then went on to produce a whole variety of different derivatives, chemical derivatives, using this cross-coupling reaction and using these 15-aryl boronic acid derivatives to give you this extra chemical derivatization strategy. Now, at that time, everyone said, well, that's great, but so what? I mean, the only point of actually making all these derivatives if they have any interesting biological activity. And it turns out that my colleague in Imperial, who sits 3 floors above where my lab is a guy called Jake Baum. And Jake's been working on the malaria parasite Plasmodium falciparum for many, many years, and has a very nice drug screen in his laboratory set up to look for inhibitors of Plasmodium growth. So Mark Wilkinson and Hung-En met each other and decided to do some experiments on this platform. This is the platform. I won't go through it in detail, but it's a very well-defined platform that allows you to measure parasitic growth, the Plasmodium growth in red blood cells. And it's actually used for the World Health Organization to screen for derivatives of Plasmodium growth inhibitors. So we took our violacin that was biosynthesized, which is shown here, and we could obviously buy violacin from Sigma, and we did some characterization work and showed that the biosynthetic violacin was actually working very, very well. And then we also looked at all of the other derivatives, and this was the first proof of principle work we did. And what was really interesting was that if you look at this graph here, so this is violacin from Sigma, this is our biosynthetic violacin, and this is 50% growth inhibition of the Plasmodium parasite in this assay. And then these are all of the other derivatives that we managed to make, not including Allen's Suzuki coupling derivatives. We haven't tested those yet. But you can see that we were getting quite a spread of growth inhibition, which was very encouraging, implying that actually what we were producing for some reason, and we don't know what the mechanistic rationale is for this, but there was some quite strong inhibitors. For example, the 4-fluoro seemed to be very good. The 5-hydroxy was interesting. There was a few others as well. And I think this gives us interesting work ahead to try and exploit what we're actually seeing here in terms of any of these derivatives being useful or not. So that was a quick whiz through quite a lot of stuff, but I hope I've told you that cell-free protein synthesis assays can be used for a variety of applications. I hope I've indicated the prototyping parts for non-model organisms, and I do encourage the build-a-cell community to explore non-model organisms as a really intriguing source of innovation and knowledge for building cells. I've showed you a little bit about prototyping some biosynthetic pathways in Streptomyces, and I've focused a little bit more on the more recent work, which was on the novel chemical entities using a combination of biosynthetic transformations and synthetic chemistry in the violacin work, resulting in some interesting potential inhibitors in an anti-malarial growth screen. So I've mentioned everyone involved in the work, but obviously Simon Moore has his own group, University of Kent, and Hung-En Lei is now in Victoria University of Wellington, New Zealand, and then these are the other colleagues who are now, whose work I've described in this very quick talk. So thank you very much.
Thank you very much, Paul. We can have questions in chat if someone doesn't have enough bandwidth, or you can unmute yourself to ask questions. So I want to start. This might sound a little silly, but I understand the reason for using non-model organisms for biosynthesis and for small molecule production, and you've shown a lot of examples for that. But what would be the reason for using it for synthetic cells? If it's kind of, it feels like it's hard enough to do it with the good old E. coli.
No, very good point, Kate. I do accept that point. I guess what I'm interested in is, you know, we seem to be focused. I guess what I'm kind of indicating is that we've got ourselves in a sort of a local minima, where we're considering E. coli as our main kind of lab microbe. And I suppose what I'm trying to suggest is that there are I mean, there are a lot of people studying different microbes, clearly, but there's a load of different microbes out there. And I suppose what I'm saying is that if we're into building cells, and obviously we're starting at complex but single cells, then I think one needs to, please just look around and see if there are other interesting properties of microbial systems that are being biochemically characterized or identified that could maybe lead into potentially a simpler, could even be more, different approach. I mean, we're very focused. I don't know. It's just a sort of a gutty kind of feeling. There's no scientific rationale for it, because obviously the most of the work has been done in E. coli or yeast, or perhaps one should stick with those. But I don't know. I'm just intrigued by the plethora of different microbes that we have.
Thank you so much. You have a question in chat. Can you see it, or should I read it?
Oh, sorry. Yeah. Okay. Was there any bacteria that you tried that you could not make tail system out? Really great question. This is from Amir Pandi. It's a really great question, Amir. The answer, actually, believe it or not, is no. But obviously, we haven't tested everything, and we certainly have only explored a small subsection of what's possible. Having said that, Amir, it's not easy. The Streptomyces cell extract optimization took around 9 months or a year. Although we had a signal to get it into an optimal version that you could use for production and biosynthesis, we spent a lot of time optimizing it. So that was a tough one. So I think, my gut feeling from my evidence so far is that you could probably make a cell-free extract from any microbial system, but the length of time to get a successful and optimized system can vary quite significantly. But I think there are enough tools out there to, and high throughput screening technologies, and we also use statistical learning as well for some of the optimization steps to really get an extract operating. So yeah, it's a very good question. Did we use automation while producing or working with cell lysates? Wondering about cell lysate batch consistency, and automation helps with that. So that's another really great question from Ben Scott. Yes, we do use a lot of automation. So a lot of this work is carried out in London Biofoundry, and we use the acoustic liquid handling system as our main lab site, as our main robotic platform, where we do a lot of optimization and a lot of statistical learning around optimization of energy sources, optimization of buffer conditions, and all those sort of things. It's very, very true that cell-free lysate productions do vary from lab to lab, and this is one of the great inconsistencies that people are fighting with sometimes. Even for E. coli, there can be enormous variations, despite all the wonderful work from Vincent and Mike's group, who have really tried to nail the variability down. I think using automation and using systematic measurement tools will help in this, and I think that's really important. And one of the projects we're working on actually with Richard Murray at the moment is to try and define some kind of tools to allow systematic comparisons of cell-free extract performance criteria, if you like. So that was a great question. So yes, it does really help. Is the hope to transfer the prototype system back into natural cells? Yes, great question again. This is from Chimeng Tan. So Chimeng, yeah, that's a really good question. The prototyping is all geared around trying to bypass, or not bypass, to accelerate the steps required to get living systems optimized for whatever particular circuitry you're designing. So yes, that is exactly the aim. Has it been absolutely proven yet that a completely in vitro defined, designed biosynthetic pathway popped straight into a living system is the most optimal version of that pathway? No, it hasn't been proved yet, but it's certainly ongoing work, and certainly a target for most people who want to push prototyping. What prototyping does allow is access to a whole lot of characterized genetic parts that may not be available for you to explore. I think that's absolutely certain now. And unexpected context-dependent issues are a key issue. Now, actually, that's one of the most interesting things about cell-free prototyping, and I would argue could actually be a very good way to use it, is to actually explore where the relationships break down. So as you can see from quite a lot of simple promoters and RBSs, at least for these very simple microbial systems, there does tend to be a relationship between in vivo and in vitro. But that doesn't always hold true. And actually, one of the interesting observations we made in E. coli a while ago was we had a synthetic super promoter that was so darn active that it was massive producer in the cell-free system. I don't think we actually published it. It was just, we didn't know what to do with it. It was ridiculous. It was just a massive overproducer. We then put it into E. coli living system, and it was one of the lowest promoters in our library. And clearly what happened there was that, you know, it was, the cell itself had actually turned down that promoter to a level that it could cope with. And, you know, you could argue what was the mechanism of that? We didn't really bother looking at it. It was just one of those weird observations. But clearly that sort of context dependency, I think, is really interesting. So where this breaks down, I think you could learn a lot, particularly in the design process, and the other way around. So I think that's a great question. I hope that answers Jim's question. So Karina, to produce different derivatives of isin, how would you go about optimizing production of a specific derivative? Would that be in vivo and vitro? Yeah, great question. It would, I think, to be, that's a really, really good question. There are a lot of people out there that would argue that in vitro, as a production vehicle or a production, you know, system, could be very competitive with in vivo, and it depends on how much you're wanting to manufacture and make. But I still feel that if we're talking about doing large-scale screening, I would probably go for in vivo. And I think with in vivo, you can get quite high yields for the optimized violation pathway. So I would plonk on in vivo for that particular application at this particular time. Can a robust cell-free system be developed from a wide array of non-standard organisms then, and comment on timeframes you mentioned? Yes, I believe it can be developed from a wide range. So I know of several other groups who are working on, including my own group, who are working on extremophiles, who are very interested in both thermophiles and also other types of extremophiles that live in high salt. In particular, we're working on one of those kind of organisms in the lab. And I think those kind of organisms are really kind of interesting for lots of reasons. But I still feel, so the vision would be to have a biobank of cell-free extracts from multiple different organisms, both eukaryotic, prokaryotic, all with kind of different kind of contexts, different phenotypes, different opportunities for you to use in different contexts. And that would be kind of like a vision I would have. So if we don't know how the thermophilic cell extracts might work, but they might be very useful in producing, proteins of particular characterization, for example. We just don't know how that's all going to work out. But I think the idea of having a bank of different organisms is a really good idea. Some people have said, why don't you mix extracts together from different organisms? But I'm still struggling with that. I guess one could do systematic mixing and see if you could get different optimizations or different phenotypes, but it's a whole area people are interested in. I wonder if there's ways to synthesize violation chemically. Yeah, so we have compared. It's a very, very good question. This is from Ankit Roy. So Ankit, we have compared that. You can get chemical synthesis of violation. I think it's I'm going to stick my neck out, and I can't remember exactly, but I think it's there are 6 or 7 step biosynthetic pathway. So it's not out of order for a good synthetic organic chemist to do. I guess the question is, some of the derivatization gets more complicated. And I think there is this kind of screening approach can be done very, very quickly. And as I showed you, you can screen rapidly analogs of great diversity at very small amounts to see if they have any biological activity quickly. And then if you identify a particular analog which you're very interested in, and you've got some really good motivation to expand the analysis on that particular analog, then of course one could look at using synthetic organic chemistry as a way to synthesize a large amount of that material. But that's a great question. And I think the lesson there is always look at existing methodologies to make sure that what you're doing is not completely a waste of time. So great question. And Kit, here's another question from Peter. Eukaryotes is recapitulate. Yep, great question again. So I glossed over that. So what we produce is a cytoplasmic HeLa extract, which can only allow the essentially translation and part of the whole process. because you end up having to put in T7 polymerase? That's a really good question. So there are a lot of challenges to reconstitute both the transcriptional nuclear activities of mammalian systems with, or of eukaryotic systems with the cytoplasmic chromatin, to making that linked transcription translation. If you think about a mammalian system, we have messenger RNA being produced within the nucleus, then being exuded out through the nuclear pore into the endoplasmic reticulum, where ribosomes grab hold of it and start doing all sorts of translation. So you have a spatial distinction between transcription and translation. So how do you couple that in a test tube? That is a big challenge, and it's some people are really interested in trying to do that. My own lab as well. And the nuclear transcriptional assays you can do, and that was done years ago, in fact, when people were studying the regulation of mammalian genes, or you're running these nuclear transcription assays in a test tube was clearly a way of identifying new genes or new promoters and characterizing them, and that works. But the activity of that is very, very different to the translational activity, and trying to couple and get both systems working at optimal levels is a challenge. So good question. I have no answers for you, but I think it's a great challenge. And it would allow you then to explore. And then, of course, you have the other problem of all the post-translational modifications that go on in the nucleosome. These are all the kind of methylation types of issues with the histone tails and all of the opening up of the chromatin and all those kind of context dependencies that you could well miss if you're just doing simple plasmids. But that is a great challenge, and one I think people should be really interested in exploring. Perhaps no question is a stupid question. Whoever sent that, you never start with perhaps a stupid question, because every question's good in my view. Can you use synthetic DNA fragments directly as inputs in cell-free systems? Yeah, you can, and that's an important question to raise, and I probably glossed over all of that. Sorry. Yes, you can. So you can take completely synthesized linear pieces of DNA and drop them into a cell-free system and get transcription and translation to occur in microbial, in E. coli systems, cell-free systems. So that really does reduce the bill phase. So that's a very good point. Do you think it would be an advantage in trying to replace pure cell-free machinery with some non-model organism protein? Yeah, very interesting question. That's from Ali. Yeah, I don't know. I mean, one thing for the builder cell community, I think, is the reconstituted pure system offers a lot of, nicety, if you like, because you have a series of biochemical components that allows you to have the system highly characterized, highly modeled, if you like. You can model that system very well. When the trouble with cell extracts is that they contain, the E. coli cell extract might contain 600 different proteins of different types and varieties and things. So it's a good question to know whether one could mix the pure system with other non-model organism proteins. Of course you could, but the pure system is so well defined now. You would have to have a very good rationale for why you'd want to do that, but it's a good question. You could explore other transcription translation machineries from other organisms in a pure context, which might give you some insights into whether that system works more robustly than the pure system, which does produce signal, but the signal is nowhere near as high as in an E. coli cell extract. I think, is that all, Kate?
I don't see any more questions on chat, and
those are all really amazing questions. Thank you so much.
Thank you, Paul. That was really, really good. I think we learned a lot about what we actually need to do to make a synthetic cytoplasm, which seems like the hardest part of making a cell.
I wish you enormous luck.
I mean, you're working on it too. You don't just
I
wish us
know,
luck.
but I don't. I know, but it's a greatest challenge ever, I think. And it's an amazing project, and it's a really great thing to focus on, because you're going to learn so much about the fundamentals, which are very, very it's really great.
Okay, thank you so much,
Great.
Paul, and
No problem.
thank you everyone else.
Yeah, thanks, Kate. Thanks everyone. Bye.
Bye.